And it's actually kind of an insane thing to think about, right? Because as a T cell, you're going to have to come up with a bunch of different keys,
Right.
right?
Right. For
And
all
that
the
means
stuff.
for all of the stuff that's out there. And if you want to have a bunch of different keys, you want to have a diversity. This is in the protein. You want different shapes in your protein. That means you need different sequences of amino acids. You want different sequences of amino acids. That means you need a different sequence of DNA creating that amino acid. And for the longest time, it was like a really big challenge to figure out, because we have like something like 10 to the 15 different receptors. Okay, to code for 10 to the 15 different receptors, you would need like 10 to the 13 amino acids at least, or 10 to the 14, right? Because it's 3 for 1. So I'm doing some really crude math here. 10 to the 14 different sequences of DNA, that's a lot of DNA.
Right.
Okay? That's larger than our genome.
Right.
Okay. So how does that happen?
How are we actually storing the memory and facilitating the creation of the T cell receptors in this wide, this massive scale that is necessary for us to survive in the way we survive?
Exactly. You've got all these different shapes, right, that the T cell receptor can be.
Okay.
Right? And it's trying to generate all of these different shapes. And the challenge is, how do you generate so many different protein sequences without blowing up on the genetic side of things?
Right, correct.
Right?
Correct.
And that was made possible by something called VDJ, variable diversity and joining locuses. They're different gene segments. And what ends up happening is the cell mixes and matches. So it's like you have like 10 different pairs of socks, 20 different shirts, 20 different pants.
Yes.
And now you can come up with a combinatorially much larger outfit.
Yes.
Right? That's exactly what these T cells are doing.
Got it.
Okay. And this process was discovered by Susumu Tonegawa, and he won the Nobel Prize in 1987. He's now at MIT. He's doing actually optogenetic stuff, which is what I had slated
as the prediction.
He's actually chasing his second Nobel, I'm pretty sure.
Oh, okay,
He's trying to. He does like optogenetics on the hippocampus, which is the center for learning and memory in our brain. So good luck to him on that.